So I'm an orthopaedic surgeon. I'm good at anatomy, I'm good at biomechanics, I'm good at cutting things and putting them back together. But really, carbon chemistry was never a passion of mine, and that's why I did orthopaedics. I didn't have to think about it ever again. But I needed to understand what was happening with ketones and what the hype was all about. So again, I try to make things as simple as possible, and we're going back to this model that we can either burn glucose as our primary fuel source or fat as our primary fuel source to get energy. So in a nutshell, this is what you need to know about ketones. The fatty acids are released from the fat cell. They travel through the blood into the liver. In the liver, there's this magic box which turns them into ketones. Ketones are released back into the blood. And from the blood, they go to the cell where they provide energy. A certain number of them will evaporate and be blown off through your lungs, and a certain amount of them will get excreted through your kidneys. Thank you very much. So where does our energy come from? When we eat food, it's broken down into its small bits that are useful to the body. And the mitochondria is the powerhouse of the cell. This is where most of the energy production takes place. A major role for the mitochondria is production of ATP, which is sort of the unit of energy. And this is used to transport stuff in and out of the cells, create mechanical changes like getting muscles to contract, and help synthesize or build new parts of the cell. So at a mitochondrial level, and that's what I'm showing you over here, is what happens within the mitochondrial. And don't get scared about all the chemistry and the chemical equations. There's two major things I really want you to notice. That both fats and glucose go into the mitochondria. Now, in a standard diet type person, about 50% of your energy comes from the carbohydrates, and about 50% comes from the fats or fatty acids. So this is people who are on a standard Australian diet. These feed into something called the TCA cycle or Krebs cycle, and I'll talk a little bit more about that afterwards. If you become fat adapted and you cut out the glucose, the amount of glucose feeding into the system gets less, the amount of fat feeding into the system gets more, and for example, after 3 or 4 days of having less glucose, your brain can adapt to use the ketones instead of the glucose. So again, complex slide, don't worry about remembering the details. This is just an enlargement of this here. There's a couple of things we need to know about. One is called acetyl-CoA, and the other is oxaloacetate. So burning glucose as our primary source of fuel is known as glycolysis. From carbohydrates, we make acetyl-CoA, which you see down here, which enters something called the citric acid cycle up here. And again, you don't need to remember this. This is more a conceptual thing that you need to understand. From which, down here, the energy is released. Now, it has to go full circle to come back again. And if you're missing one of these ingredients, it's a bit like missing a critical step in a recipe. You don't have eggs, you can't make the cake. The whole thing will fall apart. It's the same thing here. And the critical thing that I want you to notice is this thing called oxaloacetate. Now, ATP is very important, but we only have limited stores of glucose and glycogen in the body to burn. And I think people know that. We all know that muscles have lots of glycogen. But the problem is, if my leg muscles have lots of glycogen and my arm muscles have run out of glucose, there's no way of getting that glycogen from the legs up to the arms. The only organ that can release glycogen is the liver and send it to other parts of the body. So when my arm glycogen is used up, I get glycogen from the liver, but when that's used up as well, I've got no more glycogen. And the only way to get more glycogen is then to eat it. So with sustained exercise, we will fairly quickly run out of our glucose and glycogen stores. Now, gluconeogenesis is the body's ability to make its own glucose. And that's why we're not worried about the brain which needs glucose. We will make enough glucose for the brain on our own without needing to eat it. So I'm going to go through the slide a couple of times and go through it relatively slowly, because this is what you need to understand for ketones. This is the crux of the science of low carbohydrate energy production. Basically, this is what happens normally. Carbohydrates go through acetyl-CoA, go through that big circle that I showed you beforehand, and you end up with energy, or ATP. But if we cut out the carbohydrates, what happens? We don't have any acetyl-CoA to go into the cycle. So what happens? The body says, okay, well, I still need acetyl-CoA. What am I going to do? I'm going to make the acetyl-CoA from the fat. That puts it back into the citric acid cycle. Now, the problem at this stage is we haven't given the body carbohydrate or glucose. So the body says, okay, I'm going to make my own glucose by going through gluconeogenesis. But in order to do that, it uses up the oxaloacetate, because that's required for gluconeogenesis. And what does that do? It stops the circle going round, so we don't have any more ATP. Now, this is a problem. As the ATP isn't produced, the acetyl-CoA doesn't have anywhere to go, and so we get increased acetyl-CoA. What happens to that increased acetyl-CoA? Well, we can turn it into ketones, and the ketones can be used for energy. So I'll run through that one more time. You cut out the carbohydrates. That makes no acetyl-CoA from there. So we've got to get acetyl-CoA from our fat. When we don't have carbs, we use up our oxaloacetate to make glucose. That blocks off our energy pathway this way. We get an increase in acetyl-CoA, which causes ketone production, and ketones are used as an energy source. So after about 3 days, the brain gets about 25% of its energy from ketone bodies, and after 4 to 5 to 6 days, it can go up as much as 70%. So you can have a massive increase in your ketone energy supply to the brain. Now, the level of ketones or ketone bodies in our bloodstream can be measured, and this reflects the amount of fat that's being broken down. But we'll talk about that a little bit more later. At a cell mitochondrial level, the ketone bodies are taken up and reconverted into acetyl-CoA, where they can turn them into energy. So, remember our fat cell from the previous talk as well? The hormone-sensitive lipase is breaking up the triglyceride, the fatty acids are going out, and then they go ahead and go through the liver into the cell, and they're then burned. Now, fatty acid in the blood can either come from food that we eat or our own stores. If you're eating lots of fat, you don't need to use up your own stores. So you've got to be careful with how much you eat if you're looking to burn your own stores. So, what happens when the fatty acids reach the liver? Well, you remember that the cell wall has this barrier to get through. The same thing happens in the mitochondria, that the fatty acid can't get through there. Now, a lot of people are very worried about protein. A lot of people have heard of carnitine, and you don't really need to worry too much about this, but the only way that the long-chain fatty acids can get into the mitochondria is using a carnitine pathway. So, once they're in the liver, the fatty acids allow acetyl-CoA to be produced, and then eventually it goes through becoming acetoacetyl-CoA, goes through HMG-CoA, and you need enzymes to be able to do this, and you form acetoacetate. Acetoacetate is released into the blood, and some of it's converted into beta-hydroxybutyrate, but that requires energy for that to happen. And some of it spontaneously becomes acetone, which is quite volatile and can go up into your lungs, and you can blow it out through your lungs. Now, there is an enzyme that also does that, and so if you get a lot more acetoacetate, it can drive that a little bit more. So the reverse process can take place as well, that you can start with your beta-hydroxybutyrate and run back through the system to give you the acetyl-CoA to be able to produce the energy. Now, the question is, why does the liver not just use up all the ketones that it's making? because if it can go in both directions, then it should just end up in this endless cycle where it's making and burning and making and burning. But luckily, the body's pretty clever. We're missing a particular enzyme in the liver, which is known as thiolase, and that's what stops the liver from eating and using its own ketones. Now, again, simple orthopedic surgeon, when I was first trying to learn about all of this, I thought, what's a ketone? So I went to Wikipedia, as most people do, and I read this, and it just bamboozled me. I mean, seriously, that doesn't help me at all. So I then looked further into Wikipedia, and it says ketone, not to be confused with ketone bodies. I'm thinking everything that I've read in the literature is just using these terms interchangeably, ester, acid, ketone body. And there is actually a fair bit of science behind the different things. So a ketone group has a double bond with an oxygen and a carboxyl group over there, and acetone is certainly one of those ketones. And not to put too fine a point of it, but even Wikipedia puts beta-hydroxybutyric acid rather than putting beta-hydroxybutyrate as the picture when you're talking about ketones. And there's this complex pathway where you can remove a proton from it. But at the end of the day, unless you're actually a scientist, it probably doesn't matter exactly what term you're using. So if you read ketone body versus ketone versus ester or acid, it does make a difference from a scientific perspective, but from the general weight loss community, the discussions don't seem to make that much difference. So while we're talking about nomenclature, I want to briefly discuss ketoacidosis, because maybe every second week I get this question, you're going keto, oh my God, that's dangerous. And I think it's important for us to understand ketoacidosis, just to be able to tell people, look, you're simply wrong. This is not a realistic problem for us. So if you remember our graph showing fat burning versus fat storage and the amount of insulin that we have, we can see that when insulin is low on the x-axis, fat metabolism is high. As insulin levels increase, we get a sharp drop in fat metabolism. So this reflects the fact that even small amounts of insulin are going to have a large impact on our fat burning ability. This is very important for our next slide. So this is a graph of pH, which is the acid-base balance of the body, and this is the amount of ketones you have in your body. So again, just a quick show of hands. Who of these, who in the audience is ketogenic? Okay, who tests their ketones? Excellent. Now, has anybody had a ketone level of above 2, above 4, above 6? Amazing. Above 8? So we've got one person. Anybody up to 12 with their ketone levels? Give me a number. What's the highest it's ever been?
I think about 15.
15. All right. So 15 is very, very high for most people on a ketogenic diet, and it's almost always associated with something like an extended fast, 20, 30-day fasting. It's very unusual for people to get that high. The majority of people are probably going to be in the sort of 0.5 to 2 ratio a lot of the time, and more people will get higher as time goes by. So the normal upper limit of nutritional ketosis is defined as 3, but I mean, this is an arbitrary number. It doesn't really mean that very much. Starvation ketosis is often about 5, but clearly in our audience here we've got some real goers who are getting up 5, 8, 10 sort of level, and one extreme example over there. So the absence of insulin signals the body the energy shortage. So if you're in ketoacidosis, you've got glucose, but you can't get the glucose into the cells. The brain reacts saying, I need more energy. Where can it get the energy from? It can get it from ketones. So the brain sends out a signal and say, produce lots of ketones. And the ketone production goes ahead at a breakneck speed. But this can only happen in the absence of insulin. The body tries to blow off the extra acetone. You remember I showed you the pathway that it goes to the cells or some to the lungs. And so you start to breathe faster. And as you start to breathe faster, the acid-base balance of your body changes. And you become acidotic, or you have more acid in your system because you're blowing it off, and you see that as a pH drop. So the number here goes down towards 7. Then you get dehydration, and that impairs the renal excretion of the hydrogen ions, which is another acid. And so your pH eventually approaches 7. Now, I've got here moderate ketoacidosis at the level of about 13. 15, which is where you were, would put you over here. And even that is still in a safe zone if you've got even a hint of insulin. Because as soon as the insulin hits the system, what's going to happen? Your fat burning drops, therefore your burning drops, therefore your ketone production stops. So this diabetic ketoacidosis really is only a problem if you're a type 1 insulin-dependent diabetic who doesn't have any insulin on board. All of us are able to produce insulin. When our ketone levels start to get higher, the body responds by giving it some insulin to stop the fat burning and stop us getting into this ketoacidotic state. So just to summarize, a complete absence of insulin is required for ketoacidosis. So it's not going to happen to anybody on a diet where they're making their own insulin. All right, what about measuring ketones? You can do it multiple ways: breath, urine, finger prick. The finger prick test detects beta-hydroxybutyrate as opposed to acetone or cetoacetic acid. And the blood levels are measured through a test that uses this thing called beta-hydroxybutyrate dehydrogenase, and it's got an NAD+ as an electron accepting cofactor. Does that really matter? No, not really. The fact is it's a very accurate measure of the beta-hydroxybutyrate ketone in your blood at that particular moment in time. So it's obvious already that some individuals have different insulin responses, and those people will be able to have a different amount of carbohydrate to somebody else. Rod mentioned this at the beginning of the talk. Some people will be 20 grams a day, some people 50, some people 100. And your metabolism, your fat metabolism will be affected by the amount of insulin that you have. So this is an example of a food diary from one of the patients in the clinic. So you can see here initially the patient's doing pretty well. They're testing themselves at nighttime. They're running 1.1, 0.7, 0.4. They're burning fat. They're eating steak and coleslaw, egg, macadamia nuts. Now, what happens next? They have a small piece of sponge. Boom, 0.2. All right? So this particular person can monitor their blood levels and understand the food, how the food is affecting them. Now, this varies enormously between different individuals. You know, the patient goes back on their good diet, and what happens? 0.7 again. They think, oh, I'll just have that little piece of toast. Boom, down to 0.2 again. All right? So you can use the ketone testing to actually monitor your response to fat burning. You also need to understand that what we do is eat food. We don't eat ratios on an app. And the problem with following your ratios too closely is that the food combination actually creates different insulin responses. So we know that protein causes about half the insulin spike of carbohydrate, but double that of fat. And that's a lot of the reason that we get people to limit their proteins. You've got to have enough protein for you to have your building blocks, but not so much protein that you stimulate an insulin spike and stop your fat burning. So what do we consider target ketone levels? Look, this is incredibly different depending on what you're trying to achieve. For fat burning, I would say these are probably the appropriate levels. But I had a long chat to Dawn Lemaine when she was here in Australia, and she's an oncologist. And she's told me that when she runs her patients, she runs them at average between 3 and 4 while the patients are undergoing chemotherapy and radiotherapy. So it just depends on what your goals are as to what advice you're going to be given as to how to run your ketones. Now, there isn't an absolutely better level of ketones, and it's very important to understand that. Having a higher ketone level doesn't necessarily make you burn more fat and lose more weight, because it depends where the ketones are coming from. You can drink exogenous ketones and get a very high ketone level, but what that's going to do is stop the body burning your own ketones and stop your body burning fat. So having a champion, I've got a 6 level of ketones where I've drunk exogenous ketones, doesn't necessarily help you lose weight. And Paul, I'm sure, will talk to you a little bit more about that. So to lose weight, you need to listen to your hunger signals. And most of us doing this way of eating will probably only eat once or twice a day. Very rare for us to eat 3 or 4 times a day, but occasionally you will be hungry. And on those days, you will eat more. There's nothing wrong with learning what hunger is and following that pathway. So weight loss requires negative energy balance. Ketosis tells you that you're able to metabolize fat, but you do need a negative energy balance to need to metabolize your own fat. And that really is a key point to understand. So I'll say that one more time. Ketosis shows that you're able to metabolize fat, but in order to burn your fat, you're going to need a negative energy balance. So what are the metabolic advantages of nutritional ketosis? Well, one, it shows you that you're able to metabolize your fat. Number 2, it tells you your blood glucose levels and therefore your insulin levels are controlled. And number 3, it reduces hunger, and therefore it's easy to reduce your food intake. So I would commend you to becoming ketogenic if you're not, because of all of the advantages. Thank you.